Good afternoon, everybody. Uh, my name is Daniel Valentini, and I'm joined here by my fellow presenter, Vadim, who will be presenting with you after me. And we're both recent graduates from the Sejep Dawson College in the Pure and Applied Science program. And welcome to our workshop for Introduction to Python in Science and Biology. And we're going to be introducing you guys to Python, and there's going to be a question Q&A period at the end if you have any questions. But for now, I'm going to take you through some basics for how to code in Python. So before I get into actually like coding in Python, I just want to like explain a bit about like what is coding and what is programming. So those kind of mean the same thing, but in the most brief way, coding is the process of writing instructions for a computer to perform a specific task. So like as if I wanted to help you, I mean, if let's say I wanted to like get you to come into this room from the metro, I could say like turn left, come up the stairs, turn left, turn right to be able to get in here. We can write instructions for a computer to do a specific thing using code, and we can create software to do things like analyze data, visualize data, or do computations that we want to do. And like how I'm communicating right now in English, we need like a language to write this code in for our programming. So today I'm going to be introducing you to Python, which is a high level programming language, which means that it's more readable, like as in the sense of being closer to English compared to low level code. Like at the lowest level, you have machine code, which is like actual binary, zeros and ones, but there's some like layers between that. So a low level language would be something like C, which is fine if you haven't heard of it, or if you have, which is like more from like the 70s, where that would be closer to machine code. And that's a compiled language. So all of your code is compiled at once and then turned into machine code and then ran. So that's faster than Python, whereas Python's an interpreted language that's read line by line and interpreted into machine code. So, but Python is really popular in science communities because of the libraries that are made by the community that have built-in functions that people have made to do really specific tasks. And it's also a really great learning language because it's really accessible for new coders while also being very powerful for advanced coders because of all the libraries, and it's very easy to use. And it's great for research in general and specifically for bioinformatics. Some of the really common libraries that we use in science research are NumPy, that I'm going to show a bit, Matplotlib for data visualization, Pandas, scikit-learn, and a library that's specifically used for bioinformatics is BioPython. So today I'm going to be showing some introduction to Python using Google Colab. Colab, if you haven't used it before, it's an online platform that you can use to write and execute your code. And it's a bit different than other like IDEs or integrated developing environments like VS Code or something, or PyCharm, where in the environment that you're coding in. So here you can like have your independent coding blocks like run independently of one another, without affecting each other, but then they can refer back to each other, which is really helpful. So I'm going to get into the code right away. So if you're online or you're on a laptop right now, you can, if you have the Colab file, you can go to like, well, this is in French, but it should say file here, and then you can save a copy in your drive. And if you do that, wait, maybe I shouldn't have clicked that. But if you do that, it will save your own version that you can use. So now, if you save your own copy, you can save all the edits that you're doing as you go along. So to begin, I started with just like showing how to import basic libraries like the math library and the NumPy library. So something to show here that's cool is when you want to refer back to using the NumPy library, I import it using an alias, so np. So if I ever want to use the NumPy library again with a specific command, I can just do np dot the command instead of NumPy, which is just common practice in coding in general using the NumPy library. So we're gonna get pretty introductory to start, but this is like really like textbook. If you've ever learned to code or you're just learning to code right now, I'm gonna like redo it. So I'm just doing like print hello world, and this is just to make sure your code is like running and your program works fine. So there we go. We have our first line of code working. The next thing that I really wanted to show was I'm starting with already with a variable here. This variable I'm calling name, and I'm gonna just, if you're coding along, where I've written like print your name, you could put your name in there. So I'm gonna put Daniel for me. So this is, I'm actually printing it's called an F string here. So in the string that I'm printing, here I have name, and that's calling back to the value that I've set for the variable. So since I put Daniel here, this special type of F string will call back to name, and then whatever value, well, the string that I put here, it will print along with that string. So there we go. Hello, Daniel. Welcome to YBS 2024. So just some other basic printing things that I wanted to show. Using the math library that we imported before, this is how you would refer to a library to print a specific command. Like here I printed just the value pi. That right now it doesn't really do much, but you can use that to do calculations if you want or if you need them. Something else that's really helpful as you're coding along is that I want to show is example. If you want to explain how your code is working to somebody else or to yourself, it's really helpful to comment your code as you go along to explain how it's working. You can use just like the hashtag or the pound key, and you can write these as you code along. So like here I explain like integer, float, string. These don't actually affect the code I'm writing, but it's really helpful for when you have like hundreds of lines of code, you might like start to lose track of what your code does. So this is really helpful to know like what you're doing. So I started to like just introduce some basic data types here. So I made a variable x and it's just 5. So that's just like an integer, another variable y for just 3.14, which is just, this is an integer. If it has decimals, it's referred to as a float. So you can actually see the type as you're going along. So if I want to know actually like what I'm referring to here is if I use this type command and I check, let's say x. So let's see, make sure I'm not lying. So, oh wait. Oh, I put too many. There. Okay, I'm not lying. See, it is actually an integer. Or y should be a float. So, and here I just have like a variable with a name. So Johnny, doesn't really matter which name, but this should be string. So str for string. And then here I have L, which is a list. And I have the data type, the list here, But I wanted to show something interesting here. If let's say you wanted to print one of the values from the list. So here I printed the whole list, so that prints out here. But let's say I wanted to print the number 2. So you think it could be like you think, okay, I want to print number 2 from the list. But if you do 2, you actually print number 3. So what's actually happening here is Python starts the index of your list at 0. So the 0 index here, if I print the 0th like index, that's actually number 1 there. So the 0 index is number 1, and then the 1 index would be number 2, and the number 2 index would be number 3, and the number 3 index would be number 4. So here you can also do addition with your variables as you're going along. So if I set my x here to 5 and my y here to 3.14, I can get 8.14 as an output. But let's say I do 20 and then I run this again. Oh, it's still 8.14. So does anyone know why it hasn't changed yet? Why isn't it giving me like 23.14? Well, if you're still wondering, it's because you have to go back and run your previous code before you run your new code again, and then you can get your new answer. So that's something I do a lot. Like when you're beginning to code, you always have to remember to run your previous code, or else what you're doing won't actually work. Another thing here is you can combine strings with variables. So here I wrote like a name. It's referring back to, like, let's say I change this to Daniel, and then I run that again. Make sure you run it again. But then I can combine it with hello, and then with my new variable, it can combine with the plus to make hello Daniel. I'm also going to show conditionals. So just all the basic essentials for learning how to code. So here, right now, actually this example doesn't really make sense right now, but just to show that, let's say I set a value to 42. And if I say, if this value W is greater than 10, it would print X is positive. Well, let me make it make more sense. So there. Sorry, I'm just looking over here. I gotta type on the right and look on the left. There. So now W is 10. So if W is greater than 0, print that it's positive. So that's catching that, and then it's printing that X is positive. But if I would say, let's say I do negative 10. Now what happens is it's going to say if W greater than 0. Okay, it's not greater than 0, so it'll skip to, well, now we just have an else statement. So whatever else is going to happen, whatever's going to happen is just, now it's just going to go through the else. So else, it's going to print X as non-positive. But you can also create conditionals with else ifs. So here I have an example with like grade distribution. So here I just put like the grade 80, but if the grade is above 80, it'll print the grade as A. Else, if it should print above, if the grade is above 90, it'll print A. If it's above 80, it'll print B. Above 70, C, etc. But let's say I do 30, so you get a 30 on your test, which is all right, but not great. It'll go through and print. it'll check if it's greater than 90. It's no. Okay, so greater than 80? No. Is it greater than 70? No. Is it greater than 60? Okay, so it's none of these. So it goes through the else. So you get F. Another essential building block of coding is loops. So here I have the for loop, which here I put I, but for whatever in this range. So this range function will actually print 10 numbers. So if I do range 10, this function will print 10 numbers, but starting from 0. So it won't actually print 10 because it's printing 10 numbers. If you wanted it to print 10, you'd have to actually put 11 numbers. So with 0, you'd get 10. So it's going through, and for every let's say number or integer in this range, it's saying like for the first one, so 0, print 0. So for every like integer, or let's say index I here, so it's going through printing, then it loops, prints the next one, then the next one, until it finishes the range. And then I also wanted to show like a different type of loop that you can use, is the while loop. So it's cool. You can use a while loop to make like a scoreboard or a counter, because let's say you set a count to begin at 1, and you want to say, okay, you want to count up to 10. So it's kind of like two different ways to count up to 10, but sometimes a for loop is more useful and sometimes a while loop is more useful. So here I start with a count of 1, and then I want to build up to 10. So while my counter is still under 10, print the count and then add to the counter that we've already built. So this is actually called like an increment, but you can actually write this as count equals count plus 1, but this is like a shorthand to, so if I do this, this is like probably what you would do the first time you would write this. So you just do count equals count plus 1. So it starts at 1, and then while it's less than or equal to 10, it prints the first number. So it prints 1, and then it says count equals, it adds 1 to the count, and then it goes to 2. And then when it gets to 10, when it adds the 10th value, it says, okay, it's less than 10. So it prints 10, but then it adds 1, and then it goes back to the loop, but then it can't print the 11th is actually registered, but it won't print it because it's while it's under 10 to print. So actually, if you wanted to print 11, you could just print count outside of the loop. So then it actually registered to 11, but it stopped, the while loop stopped it from printing before it could actually print. So if you print it outside of the loop, you can actually see that it did register as 11. And you can also do for loops or loops using other types of data types instead of like integers. You can use strings. So here I have my favorite fruits. So I made a list. Oh, I don't even know if I've been talking this, but I made a list with apples, bananas, and cherries. So you can use a for loop to iterate over this list to print each thing. It doesn't just have to be numbers. So let's say I wanted to add kiwi, or it doesn't have to be, you can write anything. You can write your own name, Danny. So for, let's say, the fruit in this variable, I've created a variable that's holding a list of strings here. So for the fruit in this list, print that fruit. So it goes through, okay, it prints apple, then it prints banana, then it prints cherry, then it prints kiwi, then it prints Danny. So if I reprint this, it'll just print one by one as I go through. I think I got like 5 minutes. So last thing that I want to show before finishing up is I want to show one of the most important things in Python is what you can do with programming is defining a function to do a specific task. So here I have a really basic function that's just, I'm defining a function called add that takes arguments. These are called arguments. So it takes arguments A and B, and then it'll return A plus B. So that's just general, but if I create a variable sum to like hold that result, and then I use my function that I've made, that I defined called add with two numbers, it calls the add function, and then it does 2 plus 3, and then it returns it here to the variable. And then if I want to print that, it prints a sum. But you can make a function to do anything. So I can it can still be called add, but I can change it to do return A divided by B. And let's say I have, it's still called add, which doesn't make sense, but it doesn't really matter because all that matters is actually what it does. So, so I can do 10 2, but it'll return the division now. So there it prints 5, which is really cool. And a function that I made that kind of like combines a lot of the things that we did is I made a function that checks if numbers are odd or even. So here I made a first version that kind of is like kind of how you might try to code this the first time you would do it. And then I made a second version that's like, it's a bit clearer and would be better used for like programming. So here I made a first function that it takes a number from the user and it actually it casts the input, because the input function here would actually initially take it as a string. So you need to, it's called casting. You need to cast it to an integer to actually use it to do a calculation. So in my function, I check that if the number, and then it's called like the modulo operator, which sounds kind of complicated, but it's just like your number modulus 2 or mod 2 is just saying if you divide that number by 2, what is the remainder? So if I run this function that I made and I put 27, it says 27 is odd. But what it actually does is it asks for the input, so 27, then it does, okay, if this number, so if 27 mod 2 equals 0, so is 27 divided by 2, is the remainder of 27 divided by 2 0? No, because it's odd. So only even numbers have 0 as their remainder when you divide them by 2. So it skips the print if number's even, and it just goes straight to the else, and it prints that it's odd. So if you do an even number, you would get that the number is even. So here it gets picked up by the if, and the remainder divided by 2 is 0. And I made the same function, but it's a bit cleaner to ask for the input outside of the function. It does the exact same thing, but if you want to refer back to it, it's a lot easier to use. So this is a cool function that you can make pretty basically in Python pretty easily. And then you can even refer back to it after. So if I just use check odd or even, and then I put in a value, I can still check if a number is odd or even. And I think that's all for me. So I'm going to pass you over to Vadim, who's going to continue with a model for cancer.
I think we had a question from the virtual audience.
All right.
So they were just wondering if this Google Colab drive will be shared.
Yeah, it should have been shared beforehand, but if you don't have it right away, if you go to like iscb.org, I don't know if it's going to work, but you can just go to the YBS page, and it should be the first collab there. And then Vadim's collab should also be there. So on the YBS page here, from the homepage of ISCB, there's our beautiful faces right there, and Sharif's platypus sequence, and then my part 1 here, and then Vadim's part 2 here. So you can go into those collabs, then save a copy to your drive, like I showed at the beginning, and then you can play around with it whenever you want. Thank you.
So hello everyone, my name is Vadim, and today I will present the second part of our workshop, which is more focused on the applications of Python in biology. So to begin, I would like to give you a definition of bioinformatics. So bioinformatics is an interdisciplinary area of research at the intersection of biology and computer science. And it focuses on the applications of computational methods to solve complex biological problems. So in light of this definition, what kind of questions could bioinformatics try to answer? It's a question for the audience. Yes.
Did you want to speak into the microphone?
Oh, sure.
It might be faster if you just.
Yeah.
No.
Can you answer questions in medicine or protein biology using bioinformatics?
Yes. So do we have other answers? No? Okay, so these are kind of themes and questions that bioinformatics addresses. Here are more. So for example, what are the genes of an organism? What is the function of these genes? How do proteins that genes code will fold? What are the alleles that make a person susceptible to disease? And what medicine will work best to cure them? What are the organisms that live in an ecosystem? How can we study bacterial diversity and reconstruct individual genomes from a sample containing several bacterial species? What is the evolutionary relationship between two organisms? And generally speaking, what are the processes that happen within an organism? So under each question, I put some keywords that you can look up if you're interested by the question and learn more. So bioinformatics has two main approaches. The first one is data analysis, which is the use of computational methods to analyze large amounts of data that could be the patterns in which couldn't be discovered without the use of computers because the amount of data is too large. We also have simulations, which are mathematical and computational models based on empirical knowledge that are used to make predictions about real-world phenomena. And examples of questions that both can try to answer are, what is the function of a gene for data analysis? And how does a protein fold for a simulation? And as we will see, these approaches aren't very distinct. They rather form a continuum. So since I'm more interested in simulations, my presentation will be centered on this theme. And to begin with, we need to understand what is a model. So a model is a simplified representation of the world or a phenomena. And to build a model, we need to go through some steps. So first, we need to gain some knowledge. Gain some knowledge from the universe, which is represented by 1. We need to translate this knowledge into mathematical language, which is represented by 2. Then when we have equations that can be gathered together, we obtain a model, which is represented by 3. Then we can use the model to have real-world data, which is called inputs, that is analyzed by the model, which is represented by 4. And these inputs lead to outputs, which are expressed mathematically, but can be converted into the language of the real world and lead to predictions. And simulations are really parts 4 and 5. As it is represented in this picture, modeling allows us to convert qualitative information into quantitative information and back. So models and simulations have many purposes. For example, they can allow us to test new hypotheses by adding assumptions to a model in the form of mathematical equations. They can also show gaps in our knowledge. For example, when the outputs of a model won't match experimental data. And models can also help us to make new discoveries by refining an experimental design based on predictions to choose design an experiment in a way that will be more likely to make a discovery. And generally speaking, modeling is very similar to engineering because we ask ourselves a question, gather information, design a model, test it, and improve it. And it's a very creative process. So models come in two types. We have deterministic models, which don't include any uncertainty in their calculations. The same inputs will always lead to the same outputs in a deterministic model. And an assumption of a deterministic model is that under the same initial conditions, the final state of the system will be always the same. And an example of a deterministic model is the prey-predator models used in population ecology. We also have stochastic models, which will include uncertainty in their calculations. For example, instead of having a single value, a variable can be assigned a range of values. And in the case of stochastic models, the same inputs can lead to different outputs. And stochastic models assume that the same initial conditions of a system can lead to different final outcomes. And examples of stochastic models are cellular automata and artificial intelligence. So, as I said, all these models and approaches lie on a continuum. So, on one side of the continuum, we have data analysis, and on the other side, we have modeling. But different types of modeling are nearer or further from data analysis. So deterministic models, which are very mathematical, are further away from data analysis, and stochastic models are nearer. And in between, we have hybrid models, which have a deterministic framework, but some stochastic elements. For example, we can have a prey-predator model in which some variables are assigned values in a stochastic way. And we also have artificial intelligence, which is even nearer to data analysis and can be trained in a stochastic manner. So here are some common examples of simulations in biology. So population ecology uses simulations to predict the number of individuals within a population as a function of time. And the most common types of models in population ecology are prey-predator models. We also have molecular docking, which is a molecular simulation which predicts how molecules will interact together and orient themselves with respect to one another. For example, how will an enzyme and a substrate interact together to form an active complex? And finally, another example is protein folding. So one very famous question in biology is how to predict the final structure of protein based on its sequence. And recently, we have AlphaFold, which was developed. It is an artificial intelligence system which allows to predict nearly any protein structure based on its sequence. So now we'll move to the more active part of my presentation, and present you a model of cancer growth. While I won't go over the code that I used, you will be able to find it on the YBS website. So to begin with, we need to know a bit more about cancer biology. So cancer comes in two types. We have diffuse cancer, in which case cancer cells are spread within the tissue and don't have any definite edge. But we also have solid tumors, which have a clearly defined edge. There are a group of cells that grow together. And due to this fact that these cells form a very compact mass, oxygen and nutrients won't penetrate each level of this tumor in the same way. And because of this, some cells will divide and some won't. So we'll have, generally speaking, we'll have three layers within a tumor. We'll have a layer of proliferating cells, which are cells that divide because they have enough oxygen and nutrients. We'll have quiescent cells, which are cells that are dormant because they don't have enough nutrients and oxygen to divide. And we will have a center in which we'll simply have dead cells because they don't have enough oxygen and nutrients. So how can we convert this knowledge about cancer into a model? So let's assume that a solid tumor always had a spherical shape, and that all the proliferating cells divide during each cell cycle. So we can also choose our independent variable to be the number of cell cycles, and our dependent variable, the volume of the tumor. So I began with the simplest case, in which all the cells are proliferating. So the red surface, the red area and volume on the sphere represent the cells that are proliferating. So if all the cells of the tumor are proliferating, they will all divide during each cell cycle. So after a cell cycle, the volume of the tumor will double. And this gives this graph in which we can see that the tumor will grow in an exponential way. But this model has a limitation. It doesn't include any quiescent or dead cells. So to solve this problem, I decided to add a layer of proliferating cells on the surface and a center of cells which are quiescent. So in this model, I simply subtracted the volume of the smaller sphere from the volume of the bigger sphere to find the volume of the layer of proliferating cells, and added this volume to the total volume of the tumor to find the volume after the cell cycle, because all the cells in the proliferating layer will divide. And this greatly decreased the rate at which the tumor grew. But even this model has limitations. So it doesn't account for the lack of oxygen and nutrients experienced by the cells that are near to the center of the tumor. And we can imagine that the proliferating cells that are near to the center will divide slower than the cells on the surface. So to solve this issue, I added two layers of proliferating cells. So the red layer was dividing completely and doubling, while the pink layer was dividing only by half. And similarly, by subtracting the volumes of the spheres and multiplying them by the appropriate ratios, I was able to find the volume after the cell cycle. And such a model had an even slower rate of growth because now the layer wasn't dividing completely. Rather, we had one part of the layer which was dividing faster than the other. But in the real world, the diffusion of oxygen and nutrients within the tumor are continuous, while in this case they're discrete because we have only 2 layers. So we need to have a continuous distribution of division rates along this layer of proliferating cells. So to add this continuous distribution, I used an integral. And by solving this integral, I was able to find the contribution of new cells from each single layer making up this bigger layer. And this decreased the rate of growth even more. But this model didn't account for any dead cells. So due to a complete lack of oxygen and nutrients, the cells at the very center of the tumor simply die, and often we see the formation of hollow space. So to model this hollow space, I set a certain volume after which the tumor will start to shrink from inside, so it will start to lose volume. So the dark blue area represented the volume that is completely hollow, and the light blue area represented the volume that will be lost during the next cell cycle. By running my model even another time, so represented by the yellow line, I had the volume defined by the outer edge of the tumor, but this volume was smaller than the total volume of the tumor because it was losing volume in the center. And this is represented by the pink line. But once more, my model didn't account for the radial distribution of oxygen nutrients in the dying center, because in reality, we can't have a layer that simply disappears at once. Rather, each infinitely small layer around the center loses some part of its volume. So to model this, I used another integral, and I kept the dark blue area in the center. And while it is not represented, I also was subtracting the light blue area around the center. And by having multiple lists, I was able to really subtract the volume lost in the center and then subtract the volume lost around the center in this layer, which was given by the integral on the slide. And this made the rate of growth increase because now the center wasn't totally hollow, and made the model even more representative of reality. So on this slide, we can see how our model progressed from something very exponential to something more representative of reality. But even this model has some major limitations. So I choose my independent variable as the number of cell cycles. But most models will express everything in terms of time because this is the most relevant variable to treat a patient. And my model doesn't tell anything about the number of cancer cells within the tumor or their position relative to one another. It also doesn't tell anything about the real rate of division. And it's false to assume that all the cells will divide at the same time. And finally, my model doesn't consider any interactions between the tumor and its environment, for example, with a treatment or with immune cells. By addressing all these limitations, the model will become better suited to be applied in a clinical context. For example, models of cancer growth are used to choose the better treatment for a patient, or choose when it's better to start a treatment to maximize the outcome and prevent relapse. So thank you. Do you have questions?
There aren't any questions online, but I actually had a question.
Yes.
So from your previous slide, right? So are there any methods that you would use to address some of these limitations? For example, like for the second limitation where you're not sure like what the number of cancer cells or their position relative to one another is?
So this model was very simple to be digestible in the context of this presentation, but people who do modeling usually use prey-predator models in the context of cancer. So they would have a system of boxes connected by coefficients, and everything will be expressed as a function of time, and everything will also have, like, they will be looking for the number of cells that are quiescent, proliferating, or dead. And from this system of boxes, it is possible to derive differential equations. And by solving these differential equations, it's possible to find the number of cells as a function of time. And such models, they come from population ecology. And in this case, our prey will be the cancer cells, and our predator will be, for example, immune cells or a medicine. So we can even include immune cells or medicines in the model. And concerning their position relative to one another, there are models based on cellular automata. So cellular automata is like you have a lattice, so a grid, and in the grid, you will have like cells, and the cells can be full or not full. And depending on the geometry around each cell will take an action. So like using lattices and cellular automata, it's possible to predict the position of the cells relative to one another.
Okay, perfect. Thank you.